Hello guys, welcome to Gyanvarsha. Today we'll be studying about measurement of microbial growth. Okay, so microbial growth is said to occur by in two ways. Okay, they may either increase in size, or they may increase in mass, or they may increase in number. Okay, so a microbe is said to be growing when it is either increasing in size or it is increasing in number. Okay, so we can measure this increase in mass or the increase in size by different techniques, and generally we use a combination of these techniques to give the final result, to say the total growth of the microorganisms. Generally a combination is preferred over a single technique. Okay, so measurement of cell number. This measurement of cell number, there are different methods. Let's do it one by one. First, we will be seeing about direct counting of cells using the counting chambers. Okay, so generally we have three different counting chambers. One is Petroff-Hauser counting chamber, one is hemocytometer, and the other is Coulter counter. Okay, these Petroff-Hauser chambers are used generally for only prokaryotes, whereas hemocytometers are used for prokaryotic and eukaryotic cells, and generally it is used for counting the neutrophils or your RBCs, your WBCs. Okay, neutrophils are WBCs itself. So neutrophils, acidophils, basophils, the shape and size. So for those are generally done using hemocytometers. And Coulter counter is used for larger microorganisms such as protozoa, algae, and non-filamentous fungi. So how do you generally do is, we have a slide. Okay, on this slide, in the middle you have one space. See, if you see here, there's a space, so from where you can inoculate your sample, and in the middle at this region there is a grid. You won't be able to see the grid as such. You can see it only under the microscope. So first you inoculate your sample, then you put the sample on a slide, and then you have to put a cover slip, and then when you see under the microscope, you can see your microorganisms in this way. Okay, so generally the corner boxes are counted. So there are different methods to count different microorganisms. So depending upon your whatever organism you are trying to count, you'll have to follow that technique of how to count the depth of the chamber, the width of the chamber. They are generally 1 by 1 centimeter. So these all things, it will depend on the specific organism which you are looking for. Okay, so the advantage of this counting chambers is that it is easy, it is quick, and it is inexpensive. But here you won't be able to distinguish between live cells and dead cells. Like here, when you are counting, you won't be able to know if the cell is live or dead. So there may even be dead cells which you might be counting, and that might give you a wrong result. So generally we prefer going with CFU. CFU is nothing but colony forming units. Okay, it is colony forming units. In this you have two types. Okay, first you can directly perform this using serial dilutions, and then you can generally, what if you have a sample, you serially dilute your sample about 10 power minus 1 time, 10 power minus 2, 10 power minus 3, such like this, depending on your initial turbidity. Okay, and next you will plate a certain amount of your sample. So in this there's this formula. It is very important. You will find it in any exam, in every exam. Okay, so what is the formula is, say, after you serially dilute your sample, you will take certain amount, say about 100 microliters of the sample, and you plate it on your Petri plate, okay, containing some media such as nutrient agar or luria broth. Okay, luria broth agar. Okay, so you will plate, and then you will incubate the sample, and then you will see that you will be able to see colonies. Okay, these colonies are nothing but, it is said that each colony is found from a single organism. Okay, each colony is from a single organism. So you count the colonies, which is an indirect measure of the number of organisms in your sample. Okay, so the formula here is the number of colonies which you see on your plate into the dilution factor. This is your dilution. The dilution factor is written as 10 power 3. This is not given as -3, it is given as 10 power 3, and then divided by the volume of the culture plated. Okay, let's see an example. Just give me a second. Okay, so let's yeah. One second. Okay. So let's consider that you have got about 30 you have got about 30 colonies on your plate, and you plated about 0.1 ml, 30 colonies, and the dilution you plated was about 10 to the power of 4. Okay, so how you will calculate this is total number of colonies were 30 and the dilution factor was 30 into 10 power 4 divided by you have plated about 0.1 ml of the sample. Okay, so if you solve this, it is 30 into 10 power 4 into 10 power 1, it is 3 into 10 power 6 colony forming units per ml. Okay, this is very important, it's per ml. So you have about these many colony forming units per ml of your sample. Okay, this is very important. Try to practice some questions related to this. You will find it in GATE, CSIR, and all the other exams, whatever you might be trying to attempt. Okay, so here mostly it is considered that you should be getting around 30 to 300 colonies on your plate. If your colonies are less than 30, then there might be some this technique is not good for your experiment, and if you are getting more than 300 colonies, there might be some error in your result. Okay, if you follow this technique. So similarly, instead of doing this dilution, plating, and then counting, we can also do this. We can also get the CFU using membrane filters. Okay, membrane filters are nothing but they are a sheet of paper. Okay, they have a certain pore size. Okay, they have certain pore size like this. So these papers, when you pass your sample through this paper or through the membrane filter, your sample will pass through, trapping the bacteria. Okay, the bacteria gets trapped in the pores. So here you can see that here this is the beaker 1 and this is the beaker 2. The beaker 1 has your sample plus bacteria. Okay, when you are passing it through this filter, what happens is after passing it, you will get a clear filtrate having your sample, and most of your bacteria get filtered out. Okay, the bacteria gets trapped on the pores. So what you do is you take this membrane filter and put it on a petri dish with nutrient agar or any other media depending on which organism you are trying to count. Okay, the medias are specific to each organism. So nutrient agar will allow all different kind of bacteria to grow, whereas some differential media, selective media you can use depending on which organism you want to count. Okay, which might inhibit certain gram positives or which might inhibit gram negative. Depending on that, you can use the media. Then you plate this filter paper, like you will place the filter paper on the media. You will incubate the plate for about 24 hours. Again, this time depends on the organism you're trying to isolate. It's not hard and fast rule that you have to incubate it for 24 hours. Your organism might take about 48 hours to grow. So again, you'll have to wait for that much incubation time. So you have to incubate, and then you will be able to see it colonies, bacterial colonies. From this again you can calculate the CFU for this. Okay, so this is one more technique where you can count the number of bacteria. Okay, the growth here is being measured along as in the form of bacterial numbers, increase in the bacterial number. Okay, so these are different filter papers placed on different media depending on what. Here they are trying to isolate fungi or yeast. Here they are isolating different type of coliforms or different bacteria. Okay, so this is one more technique where you are able to count. So for number, what we have seen is to count. So till now what we have seen is growth of bacteria can be seen as increase in cell size or cell mass, and also by increase in their number. So first we are seeing different techniques which are used to measure the cell numbers. Okay, so first we saw different counting chambers. Okay, here the disadvantage was we might be seeing even dead cells while counting. We might be even counting dead cells. So next, the improved version is trying for CFU. CFU is colony forming units. So this colony forming units, again the formula is very important, and you can count it using membrane, either the serial dilution and plating, or you can directly pass your sample through membrane filters and count the bacterial numbers. Okay, so next, measurement of cell mass. Okay, measurement of cell mass. Okay, mass is nothing but weight. Okay, so so directly we can calculate the dry weight of the microorganisms. It is the most direct approach to get the cell mass. What you will do is here, if you are having a flask, okay, you're having a flask. So what you do is you you inoculate your sample, let it grow, and then once it becomes turbid, you'll take your sample in a falcon or in an Appendoff. You'll centrifuge it. You will discard the supernatant. Okay, you will discard the supernatant. Then you will wash either with saline or your water to remove any nutrients or any some what remnants of your to remove any remnants of the media, and then then it is washed, and then it is dried using an oven, a hot air oven mostly, and then it is weighed. So when this is weighed, first you have to weigh the empty falcon. Okay, and then subtract the weight so that you get the weight of the microbial mass. Okay, so directly you can get the microbial dry weight. Okay, this technique is generally used for fungi. It is very time consuming and not very sensitive, so we generally do not go for this if this is an important step in a particular experiment. So another technique which is mostly very reliable is determination using light scattering, and this is done using colorimeters or spectrophotometers. Okay, you might have heard about these. So these are mostly used for measuring the bacterial mass or microbial mass. Okay, they depend on the fact that microbial cells scatter light that strike them. Okay, this technique depends on the microbial cells. They scatter light that strike them. Okay, the amount of scattering, how much scattering you see, is directly proportional to the biomass of cells. Okay, the scattering is directly proportional to the biomass of the cell present and indirectly related to the cell number. Okay, so here you can see that this is a spectrophotometer. Here the lower scale is the absorbance and the upper scale is the transmittance. Okay, transmittance is the light which has directly passed without hitting anything. Okay, so you can see that this cuvette is less turbid. Okay, this is less turbid. Less turbid means there are lower number of organisms. Here it is more turbid. There are more number of microorganisms. Okay, so you can see that when the organisms are less, the absorbance is less and the transmittance is more. Okay, when it is less turbid, absorbance is less and transmittance is more, whereas when it is more turbid, you can see that absorbance has increased and transmittance has decreased. Okay, so this is how you measure the microbial mass using spectrophotometers. Okay, you can just go through this. Determination of microbial mass by measurement of light absorption. Okay, as the population and turbidity increase, more light is scattered and the absorbance reading given by spectrophotometer increases. Okay, the spectrophotometer has two scales. The bottom scale displays absorbance and the top scale gives the percent of transmittance. Okay, as the absorbance increase, the percent transmittance decreases. So this is about increase in cell number. If you have any confusion related to any of these, or if you want to see a practical how we do this in practical, please let me know so that we can make a video for this. And if you have any questions related to this, please give it in the comments.